Hello everyone, my name is Xiangzi Li. I am a PhD student at UT Dallas. I'm excited to present my work, Scientific Discourse Tagging for Evidence Extraction at EACL 2021. As an overview of my talk, in this work we present a novel tagger for scientific discourse tagging task. More importantly, we show the benefit of applying scientific discourse tagging to two downstream tasks, claim extraction and evidence fragment detection. Let's get started. In this work, we focus on research papers in biomedical domain. As we know, papers that describe original experimental work provides a crucial raw material for all other subsequent scientific research. However, the drastic growing number of scientific literature is difficult to directly use efficiently. Automatic information extraction from biomedical literature is an important step to help researchers to achieve this goal. Traditional information extraction focuses on the extraction of entities, relations, and events, but do not distinguish the factual information obtained from prior work and the evidence derived from experiments that the author used to sell the claims. Therefore, it's important to recognize rhetorical components of scientific discourse, which distinguish observations and implications, or claims and hypotheses. We refer scientific discourse tagging as a general task that tags clauses or sentences in a scientific article with different rhetorical components of scientific discourses. The figure on the left shows an example of paragraph labeled with discourse tags from SciDT dataset. Table on the right shows the corresponding definition of the discourse tags. As you see in the example paragraph, clauses are colored by discourse types. Paper authors usually follow a logical flow in their text. They first state some known facts. Then they introduce their experimental methods that serves a goal. Then they describe the experimental results and finally make implications based on the results. In order to capture this logical flow, we formulate the scientific discourse tagging problem as a sentence-level sequence tagging problem. Here, a sentence can be either a clause or sentence. Each example is a paragraph. We propose a novel scientific discourse tagger that leverages SciBERT embedding, which is a BERT embedding trained on scientific corpus. We use a novel attention mechanism that uses an LSTM to compute the contextualized attention weights that performs weighted summation on the word embeddings to obtain the sentence embeddings. Finally, we use a classic bidirectional LSTM CRF model as the sequence tagger to tag each sentence. We use three different datasets in this experiment. We first extended the SciDT dataset. This dataset is derived from pathway logic and intact databases. It's a small dataset only containing 634 paragraphs. The second dataset is PubMed ICT dataset. It's a large dataset, and due to our limited computational resources, we take the smaller version with 20K abstracts. Each abstract comes with section heads as labels. The third one is Coda 19, which contains 10K human annotated abstracts. It is a subset of Cord 19, which is a large collection of papers about COVID-19. The figure shows the label distribution in each dataset. We compare our scientific discourse tagger against our prior work, Dasigi et al. 2017 on SciDT dataset. To show the strength of our model, we also compare against a few strong baselines on PubMed RCT dataset. We obtained state-of-the-art performance on both datasets. As ablation studies, we compare different types of embeddings and attention mechanisms. BioGlove is glove embedding trained on biomedical corpus. BioBERT is another BERT model trained on biomedical texts. We also show that using an LSTM to compute the contextualized embedding for summarizing word embeddings to sentence embedding outperforms using RNN or simple self-attention without contextualization. The confusion matrix above is Dasigi et al.'s prediction on the SciDT test set, and the one below is from our prediction. As you see, our model performs significantly better on predicting problems and hypothesis tags. We also use CODA-19 dataset to explore the transferability of our scientific discourse tagger. Our tagger can easily beat the baseline model implemented by a Cybert model. What's interesting is that we also beat the baseline by using our model trained on SciDT dataset or PubMed RCT dataset to perform zero-shot predictions. We use the mapping shown by the lines in the figure on the left. This result shows our model's strong transfer learning ability from one dataset to another. Next, let's move to the second half of our work. We show the benefit of applying scientific discourse tagging task to two downstream tasks, claim extraction and evidence fragment detection. Claim extraction task identifies claim sentences in an abstract. The figure on the right is an example abstract, and the bold sentences are the claim sentences. The task is essentially a binary sequence tagging problem. The dataset contains 1,500 human-annotated abstracts from Medline database. The baseline model uses the average pooling of glove embedding and the bidirectional LSTM CRF model. It is pre-trained on PubMed 200K RCT dataset. Our solution directly applies our scientific discourse tagger to claim extraction. We show that with PubMed RCT pre-training, which is a type of scientific discourse tagging task, there is more than 3% of absolute F1 performance improvement. The next downstream task is the evidence fragment detection. In biomedical papers, experimental figures have a central role that delivers experimental results as the evidence for the author's claim. Usually, you will have a rough idea about the entire paper if you quickly skim through the figures. We refer the evidence fragment as the text section in the narrative surrounding a figure reference that directly describes the experimental figure. The figure on the right shows an example paragraph that contains multiple evidence fragments. Evidence fragment detection task maps the text and the figure that it semantically refers to. The task is challenging because the figure reference varies, such as figure 1C, 1D. The number of target figures for each clause also varies. Even worse, the small dataset only contains 210 paragraphs. As a solution, we make a block-based assumption. In most cases, clauses in a block, which is segregated by the red lines in the figure on the right, share the same figure codes. Within each block, each figure code is explicitly mentioned at least once. Therefore, once we extract the figure codes using regular expression templates, we only need to determine where each block starts and ends by tagging BIO tags to each clause. The decoding is also intuitive. Given the predicted BIO sequence and extracted figure codes, we can easily reconstruct the labels of evidence fragments. Due to the small size of the dataset, we use a feature-based CRF model as the sequence tagger. Our results shows that using scientific discourse tags as a feature drastically improves the tagging performance, and our model outperforms the rule-based baseline solution. Finally, I want to discuss the idea of using evidence fragments to assist the writing of scientific papers. We previously showed that scientific discourse tagging facilitates the extraction of evidence fragments. We suggest that evidence fragments can be cataloged, indexed, and reused as independent documents, as an additional piece of evidence along with the traditional citations, figures, and captions. Ultimately, we hope to dramatically increase the amount of primary evidence used to generate individual claims and therefore improve the quality of those claims. To wrap up, we present a novel tagger with SOTA performance for scientific discourse tagging, and show that the tagger's strong transferability to other datasets. Then we show that scientific discourse tagging task facilitates its downstream tasks, such as claim extraction and evidence fragment detection. Finally, we discuss the usage of evidence fragments for producing higher quality claims. That's all for my presentation. I want to thank my advisors, Professor Nanjun Peng and Dr. Gali Burns. We started this work at USC ISI. This was my main work during my master's program. We also thank NIH for the fundings.